Today we're going to talk about transcription from DNA genomes, and we're going to use some of the information that we learned on Monday, especially as it relates to genome structure and open areas. And we have a lot to cover, and I'll try not to confuse you too much, and hopefully the notes and the slides are sufficient to help you get through. The text is a wonderful source of information. Of course, there's an awful lot of information there. But if you feel confused, feel free to come and find me, and we'll talk about it. So what is it that viruses have taught us about transcription? Well, they've yielded fundamental information about control signals in terms of what is required to transcribe a specific gene within a DNA template. They've allowed us to define what constitutes a promoter, the region, the sequence elements within a gene that are necessary for initiating transcription. They've informed us about enhancers, marvelous little DNA sequences that can be placed almost anywhere within a virus genome or within the host, and that function at a distance. They function orientation independently, and sometimes they're cell-specific, and sometimes they're universal in terms of their mode of action. But most importantly, they've taught us about how RNA synthesis is initiated and how it's regulated. And those are important components in virus replication, cell differentiation, and growth and development. And what are some of the important aspects of transcription? In a virus infection, it's usually one of the first events that follows infection. The virus genome finds its way to the nucleus, in the case of every DNA virus except for the pox virus family. And the virus chromosomes, as we spoke about on Monday, exist in a variety of template conformations. The papillomavirus families are those supercoiled circular DNAs that come into the cell wrapped in histones, because the virus genome itself is wrapped in histones in the virion. The adeno and herpes virus particles contain DNA which appears to be mostly naked, although there are polyamines and some other host constituents that are associated with DNA. And they come into the host cell as linear molecules, and the adenovirus genome remains linear, and the herpes virus genome circularizes shortly after infection. The human immunodeficiency virus comes into the cell as an RNA molecule, is reverse transcribed, as Dr. Racanello taught you, and then it inserts itself into the chromosome. And it's the chromosomal DNA copy that is transcribed as essentially a single gene. So it's present in one copy, whereas things like SV40, adenovirus, and herpes viruses are present in many copies, sometimes as many as hundreds of thousands of copies of molecules per cell. They've taught us that regulation and virus gene expression is strictly defined, and that they are controlled by virus and host proteins, and they are controlled in a temporal fashion. That is, the genes aren't all transcribed as once, as you see with something like a picornavirus. Rather, there is a systematic, controlled expression of gene expression. Regulation is generally controlled by the initiation event, how the polymerase enters the promoter. And although there are instances where elongation is a rate-controlling step, this is rather rare. When we talk about HIV replication in the context of infected cells, we'll tell you about some of the strategies that are used to control elongation. It's a multi-step process, and there are other opportunities for control. There's assembly of the RNA polymerase holoenzyme on a promoter, and a variety of other transcriptional machinery that interacts with the promoter that facilitate entry, that facilitate initiation, and that control the specificity of initiation. And there's always termination, and both polymerase and RNA are released from the DNA template during termination. There are some generic steps in transcription hold for viruses and also for the cells that they infect, their hosts. First, you have to find what you're going to transcribe, and that's the purpose of the promoter. It's also the purpose of a sequence of DNA that's frequently called the initiator region, which is usually downstream of the promoter and to which the promoter does not directly bind. There's a formation of a pre-initiation complex, and this is much like what we talked about on Monday with DNA replication. First you find it, you set things up, and you set them up for the initiation of RNA transcription. And there is site specificity in terms of where these molecules assemble and where they begin to synthesize RNA, usually about 24, 25 base pairs from the formation of the pre-initiation complex. Elongation, an important event. In fact, most transcripts are aborted. Very few of the transcripts that actually start at a promoter make their way through. So the processivity of the enzyme is really fairly weak, and it involves specific changes and events that occur to the RNA polymerase, and we'll discuss that very briefly. And finally, of course, termination, recognition of specific sequences within the DNA template that allow for the RNA polymerase to fall off and polyadenylation to occur. So what happens to RNA transcripts as they're being made? Well, the first thing that happens shortly after synthesis begins is that they're capped. And that's the addition of this head-to-head guanosine moiety, which has a methylated end. And that blocks and protects the end of the messenger RNA. As they're being transcribed, they make their way down the template. And when they finish, they get polyadenylated. And during the course of polyadenylation, splicing begins if the message has intervening sequences. Sometimes these messages are also edited, where actual RNA sequences are changed as a result of action of specific cell enzymes. Now, this is all occurring in the nucleus. And this is not yet a messenger RNA. Rather, it's a transcript, because it's being processed in many different ways. But these transcripts contain sequences that are recognized by specific host proteins, termed exportins, and they get transferred from the nucleus into the cytoplasm, where they become messenger RNAs and get translated. Things that regulate the lifetime of a RNA are critical. So the half-time of a messenger RNA is extremely critical in terms of how much product will be translated from that message. And some RNAs are silenced. They're silenced by microRNAs or by other cell machinery that we don't understand in great detail, but that leads to degradation, controlling the decay rate, and sometimes translation is inhibited by these silencing RNAs. So a lot of different things can happen to RNAs, and they can sit out in the cytoplasm for quite some time. There are three well-characterized host polymerases: Pol-1, which is involved in pre-ribosomal RNA synthesis, and it's not known to be used by DNA viruses; polymerase II, which is used to make messenger RNAs and some microRNAs; and polymerase III, which is used to make VA-RNAs, Epstein-Barr virus, small RNAs, and some other microRNAs. Now, the question is, how does the virus subjugate the host? What is it that the virus does that allows it to be recognized by the host? And there are a lot of different aspects to this that have to be considered, but the first thing is that the virus nucleic acid, its genome, has to get into the cell. Dr. Racanello showed you that hepatitis B virus has to complete its genome first. And that's an important constituent in terms of its being transcribed. And that transcription process is important for making the first batch of messenger RNAs and also for making the template that's involved in being copied to make genomes. Adenoviruses and polyomaviruses enter the cell. They enter the cell as genomic DNA. Adenovirus has that protein on the end of it. The polyomaviridae are essentially chromatinized circular molecules with that small open region around the origin. And that region around the origin plays an important role in their transcription profile. Herpes viruses do it a little bit differently. They introduce within the virus a virion-associated protein. In fact, there are a lot of virion-associated proteins. But one of them in particular plays an important role in initiating the replication cascade. And of course, the retroviruses have to be different. They enter as plus-stranded RNA, become double-stranded RNA, finally need to get integrated before they're actively transcribed. So there are many steps along that pathway. What we're going to talk about in terms of transcriptional programming is mainly regulation of synthesis. How is it regulated? In most viruses, there is a control that regulates timing, when specific messengers are made, and abundance, how much is made. This is fairly well understood when messages are made. Why various amounts of specific RNAs are made is still not clear. Why do you need to do this? Well, some gene products are toxic, and if they're made in large amounts at early times post-infection or misregulated, what you're going to do is essentially kill the cell. And that's what happens when things go awry. You can think about it very simplistically in terms of what are the steps in transcription of pre-messenger RNA, also sometimes known as heterogeneous nuclear RNA, and that is recognition of a strand of virus DNA, the promoter, by the host transcriptional machinery. And here it's a single purple blob, but it's many, many different proteins and many complexes of proteins that assemble together. Many of these initiation events abort. And what happens is small transcripts are made and just get released from the template. But some make it through and they begin to elongate. And as the polymerase moves down the DNA, the RNA gets fed off, it gets capped. The polymerase reaches a site that calls for termination, and both the polymerase and the RNA get released, and the polymerase goes back to find a template again. This is a schematic that demonstrates what happens to RNAs that contain introns. They get transcribed and capped, and then elongated, and the entire message is read out. Then the RNA is cleaved and poly A is added, and poly A addition is the signal for splicing. So if the messenger RNA contains an intron, that can be removed at that point. Not all intervening sequences are removed, and some messages come out into the cytoplasm as exon, intron, exon, and those get translated, but the protein that's made is usually only from the 5' exon. When the two parts are joined, that is spliced together, they get exported and translated. Now, one of the things that is interesting is that these two messenger RNAs are actually handled by different machinery in the cell. So things that have intervening sequences get removed by one family of exportins, and things that don't have intervening sequences anymore get removed by another. And in fact, RNAs that are transcribed without intervening sequences have yet another route of exit from the cell. So these things are all being recognized by different host machinery. Within the promoters of various genes in viruses are a variety of control elements, specific DNA sequences that regulate initiation, that regulate identification of where to begin, how much. And these are frequently called core and distal elements, and we'll get to that in a little bit. And the core elements provide information for recognition of the assembly site. Where does the polymerase sit? There's a sequence that's present in almost all virus promoters called the TATA sequence, and that's a defined sequence that's recognized by a complex of proteins called transcription factor 2D. So that just represents a fraction off of a gel, but it's many proteins. And this signal sequence is about 24 to 28 bases upstream of where RNAs initiate. There is an initiator which does not refer to the site at which RNA initiates, but rather a DNA sequence that helps to define where the polymerase sits. And that specifies accurate starts. And then further upstream from the core elements are the distal elements. And these are sites that sit for activating proteins and co-activating proteins. Sometimes these are viral, sometimes these are host, sometimes these are both. And finally, anywhere from within a gene to thousands of base pairs upstream, as many as 100,000 base pairs, are enhancers. DNA sequences that are position and orientation independent, and they can be tissue-specific or universal, and what they do is they enhance initiation of transcription. So here's a schematic view of the promoter as I've defined it. The TATA sequence, which is within 20 to 35 base pairs of where RNA synthesis starts, initiates. That's the core promoter. That's just a basic structure to which RNA polymerase can bind, but tends not to bind as efficiently as it might. A promoter contains the core promoter plus local regulatory sequences. And these are usually sites for binding of host proteins, and these facilitate entry of molecules and initiation. And finally, there are distant regulatory sequences known as enhancers and also silencers. So sequences that can actually attenuate transcription and transcription initiation. And sometimes these two sequences, both of these sequences, are present in a gene, but they depend upon the cell in which the virus is infecting for whether or not one predominates over the other. Now, what about the template? It's very rare that the template comes in by itself. It's frequently coated by host proteins and accessory proteins that the virus elaborates or brings in with it. But it's the first events that are transcribed from the template that lead to early gene expression. So early gene expression is the first thing that occurs in the cell after the virus genome enters the nucleus. So you get into the nucleus, you produce this recognizable template, and that leads to transcription of the first wave of virus genes, the early genes. Later, you replicate genomes, as we discussed yesterday, and that increases template number. And that causes a lot of interesting changes. What it can do is overwhelm the host, remove proteins that are involved in suppression of transcription, and the host has a whole bunch of those, and allow free genomes to be transcribed. So DNA replication plays an important role in regulation of late transcripts that are required for assembly of virions, structural gene products for the virus. What does polymerase II do? It's a large complex assembly, frequently called a holoenzyme, which represents about 30 or so different proteins. But its role is to specify accurate initiation at the promoter by recognition of the promoter, and it doesn't recognize the promoter by itself. Its other role is to respond to host and virus proteins that help to regulate the abundance of message by controlling initiation. And of course, it's important for it to synthesize RNA transcripts. Again, what are the steps in initiation? Recognize the core promoter, formation of a stable closed initiation complex. And this is just like DNA replication. This is what polymerases do. They find a site in a particular genome, they identify it, they form a stable complex at that position, and then they look to initiate whatever process it is that they're responsible for. And in this case, it's transcription. A lot of other proteins come along, and this leads to the formation of an open initiation complex, again through the use of helicases and ATP. And then the polymerase is in a situation where it can escape from the promoter. And escape is regulated by phosphorylation of the carboxy terminus of the RNA polymerase. You then get promoter clearance, elongation of the nascent transcript, and movement of the polymerase complex down the genome. There's a defined order of binding. Please don't memorize any of this. Just think about it. Refer to the cartoon, not to the words. And what we see is that this TF2D, which is composed of something called TATA binding protein, remember the TATA box, transcriptional activation factors, binds and bends DNA. In the process of bending DNA, it's opening it. The TATA box recognizes, TATA box binding protein recognizes the TATA sequence in DNA through the initiator. Another series of proteins come in, such as TF2A, and that facilitates binding of this TATA box protein to DNA, makes it a tighter complex. A whole other series of proteins come in, and they deliver RNA polymerase to that site. It's then that a final series of transcription factors, 9 proteins with ATPase activity and a helicase, come in, and they open up the promoter region for transcription. So you've got this very concerted order of binding. You then have opening of the complex, and you go from a closed complex to an open complex, and you can now initiate transcription. So let's look at it in terms of detail. Let's look at it initiation. Again, you have the initiation process, recognition by TATA binding protein and the transcription activation factors, the bending of DNA, allowing the entry of RNA polymerase, and the formation of the closed initiation complex. The next series of proteins come in, and their role is to unwind polymerase. And yet everything remains in contact with the DNA at the promoter site. But you begin to open up the DNA. And this is the open initiation complex. Nucleotide triphosphates come in, and these are used to begin to elongate RNA, and many of them form these abortive transcripts that I talked about. But when the carboxy terminus of the polymerase is phosphorylated as a result of entry of other proteins, you get release from the protein, and you get true elongation, and that molecule moves down the genome to make nascent RNA. Now, there are a bunch of assays that are commonly used for promoter activity, and they vary in terms of what it is that they measure. There is pulse labeling, there's pulse labeling in vivo, where we add radioactive nucleotide triphosphates, and we ask them to be incorporated into nascent RNA. We can then measure the amount of RNA that is derived from a specific transcript by hybridizing the product back to a DNA template. You can use RT-PCR to look at the total abundance of a message. And those measure different things. One is measuring essentially rate of transcription, and RT-PCR measures abundance. We also frequently use runoff assays, and some of these were first developed by Jim Manley here in the Department of Biology. And these are assays where we take a specific DNA template, usually in the form of a plasmid. We make it into a linear molecule, and we add back extracts from cells, either infected or uninfected, and we ask how much RNA is made. And the assay that is most prevalent today is a reporter assay, in which DNA constructs are transformed into cells, and they have a reporter on them, a molecule that you can measure, luciferase, and you can measure luciferase activity, beta-galactosidase, and you can measure beta-galactosidase activity, and things like that. The problem with this assay, though, is that you put in hundreds of thousands of templates into a cell. Only a portion of them get from the cytoplasm into the nucleus, but in many ways you're overwhelming the cell in terms of templates. And the other thing is that they're not in the natural configuration of the virus, and yet they still provide a useful assay. So these are two of the assays that we've talked about. This is the runoff transcription assay, where DNA is linearized. You add XTPs and transcription components and radioactive triphosphates, and then you measure the product on a gel by gel electrophoresis. You purify the RNA that's made, you electrophorese it through a gel, and you ask how much nucleoside triphosphate is incorporated into that band, and that's a measure of relative transcription rate. The reporter assay, which is a lot easier to do and a lot more fun, involves taking all of these chimeric genes, which can contain a promoter of interest, to which you attach your particular reporter. You introduce the DNA by transformation into cells in culture. You wait. These molecules don't replicate. They're just DNA molecules in genomes. And then after a period of time, you make an extract. You add luciferin in this case, and you measure luciferase activity and ask how much was made. It's an indirect measure because you're not measuring the RNA. Some transcripts are more efficiently translated than others. Well, there are other steps in transcription that are important. Regulation of abundance. It can be through initiation. It can also be through the half-life of a message. And mostly it's through things that we really don't understand. It could be through the availability of the template, the promoter that's used to drive transcription. Sometimes it's because coactivators, proteins that are involved in accelerating, controlling the abundance, helping the polymerase find the promoter, are modified. They can be modified by phosphorylation, methylation, acetylation, and ribosylation. So there are many different ways in which one can control the way coactivators work. And finally, we have the sequences that I talked to you about before, the role of enhancers, and the enhancers involved in changing the rate of initiation of transcription. After you transcribe, you find that many virus genes get spliced. And this is great for the virus because it allows for economy of genetic information. You can take the same transcript, you can splice it in different ways, and generate different protein products. So nuclear RNAs go to messenger RNAs. The nuclear RNAs are by and large larger than the messenger RNA for many of the viruses that we talk about, and they form what we call heterogeneous nuclear RNAs. They have 5' caps and 3' poly-A's. In fact, in adenovirus, all of the late messenger RNAs map to the same promoter. That is, there's only a single messenger RNA made, and it encodes about 6, if I remember correctly, different virus proteins, most of which are structural in nature. So how do you get small RNAs from big RNAs? And that's by splicing. So all of the adenovirus late RNAs have 4 parts. They have a common 5' terminal tripartite leader and a body. And the body is what specifies the gene product that's made. So let's take a look at this. In the major late promoter for adenovirus, and that's right here in the adenovirus genome, we see that you get a single large transcript that accumulates in the nucleus. And you can see from here that there are at least 5 and perhaps more different proteins encoded by these regions L1, L2, L3, 4, and 5. But at the 5' end of every major late promoter directed transcript are the sequences L1, L2, and L3. And these stand for leader 1, leader 2, leader 3. And these DNA sequences are transcribed and subsequently spliced together. So the intervening sequences here are removed. The leader sequences do not encode any protein. Rather, they play an important role in translation. And from these transcribed sequences, you then subsequently go out and remove one or all of these various different encoding sequences. So there are 8 transcription units in adeno, 1, 2, VA is 3, this is 4, 5, 6, 7, and 8. Some of them are early. They're denoted by E, E1, E2, E3. And some of them are a little bit later, such as this E2 protein, which is essential for DNA replication, and the DNA binding protein, and the pre-TP, that molecule that's used to initiate DNA synthesis. And then there's this curious guy down here, 4A2, which is synthesized during the not immediate early phase, but the early phase, and that's essential for directing the polymerase back to the major late promoter and getting it to synthesize late RNAs. Now, these 2 experiments, which demonstrated the fact that all major late adenovirus RNAs contain a common 5' end, were rewarded with a Nobel Prize, one to Rich Roberts for this experiment, and one to Phil Sharp for this one. So let me tell you what this is. Rich Roberts isolated RNAs that hybridized to the gene that encoded the hexon, the major capsid protein. And then he investigated the 5' end of these RNAs, and what did he find? That it had a specific sequence. He then looked at other late RNAs, and he found that they contained the same 5' sequence. So the 5' sequence was conserved. The 5' end of all of the late RNAs had the same undeconucleotide sequence. Phil Sharp took adenovirus DNA, shown in blue, hybridized it to hexon RNA, and found that at one point he got a double-stranded structure. And when he looked at the 5' end of this transcript, what he found was that the DNA looped out, A, B, and C. And these regions correspond to the regions between leader 1 and 2, 2 and 3, and where the RNA gets spliced from 3 to hexon messenger RNA. So these were the first experiments that demonstrated that RNA was spliced. And they were critical to our understanding of how viruses use genomic information to conserve genomic information to make proteins. So splicing is value added. Introns provide numerous sites at which RNA sequences are broken and rejoined. Splicing, yes?
Is that 5' sequence for the late RNAs only in the adenovirus, or is it found in all viruses?
No, it's only in the adenovirus. It's in each adenovirus, and there are, I don't know, 35 different ones that can infect you, have unique 5' ends for the RNA, for the major late RNA products. Splicing occurs without loss of coding information, so it's very economical. And alternative splicing creates new functional genes by rearrangement of exons. So here's a picture, a schematic of what happens during adenovirus transcription. And the only thing that you really need to focus on is the fact that from this major late promoter, you get this large transcript, and it can be spliced at any one of a number of sites. And when it gets spliced in part because you have various poly-A addition, but mainly because it's recognition of intron-exon boundaries. And from that, you get all of these different gene products. Retroviruses do it too. They do it in a much more complicated way. You have a small 10 kilobase genome, and this is HIV, that gives rise to a variety of different proteins, all of which play integral roles in the replication and latency of this virus. And we'll talk about some of these transcripts and the proteins that they encode and what their function is when we talk about persistence of virus infection. Now, what about enhancers? SV40, that little DNA molecule that I told you about on Monday, is a member of the polyomavirus family. And contained within the origin of SV40 is an enhancer sequence. Invariably, it's a duplicated 72 base pair sequence, and it's present in the open euchromatic region of the SV40 chromosome. You can take this sequence and move it from that region, any place in that genome, and it still functions. That was one of the first experiments that was used to define enhancers. In fact, you can take that sequence and move it to another gene and introduce it into a cell and demonstrate that it's an enhancer. It's orientation independent. It doesn't care if it's going frontwards or backwards. It operates in either context. And most interestingly, it can work in trans. And we'll show you an experiment that demonstrates that. So it doesn't have to be a contiguous part of the molecule, and that's because of the way enhancers function. So here's a structure of this 72 base pair repeat. And you can think of this as the origin, a larger sequence within which this is embedded. And what all of these little boxes represent are sequences that are recognized by host proteins. So the polyomaviridae come into cells, and they expose themselves to host proteins. And these host proteins are important regulatory transcription proteins that are involved in initiating and accelerating transcription from these genomes. Now, how do enhancers work? Well, I told you that they work in trans, and the reason that that works for us is that what happens is that DNA actually bends, and it bends such that the enhancer is juxtaposed to the complex of proteins that are involved in first recognizing the promoter, the initiation guys, the guys that are involved in forming the closed complex. And that is what brings rapidly the promoter into context and allows for the initiation complex to form more rapidly and more abundantly. Now, how do we know that enhancers work in trans? So this is a very clever experiment that was done to test that. In fact, it was done with SV40, and what happens is the entire region that contains the 72 base pair repeats gives a relative transcription rate of about 100. If you cleave it with a restriction enzyme such that you separate the core promoter from the upstream control region, that adjacent upstream region, then you get 1% of the activity. So this sequence, just the core promoter, is a relatively weak promoter. What it says is that it's the enhancer sequence and the proteins that are recognized within this region that are responsible for increasing transcription. So what was done next was to take these digested DNAs and to add a biotin moiety, a very sticky protein. And biotin binds streptavidin voraciously, incredibly tightly. And what you could do then is to bring these two molecules back together by adding streptavidin. And when you add streptavidin, you now have this sequence, the core, and this sequence, the enhancer-containing sequence, linked together. Now, of course, sometimes you get these two guys linked together and these two guys linked together. And that's probably why you don't restore 100% of the transcription, but you go from a relative transcription rate of 1 to 58. And this experiment demonstrates that the enhancers can work in trans because they're physically separated. It also demonstrates, because of the presence of streptavidin and biotin, that this is not an entry site for polymerase. That what it does is enhance the polymerase, finding the appropriate core initiator. Now, what do the host proteins do? Well, the host proteins, of course, recognize the virus promoter, and their role is to, at first, regulate early gene expression. Viruses can either encode or bring with them co-activating molecules, and that helps the host find where it needs to be. But cell type specificity can limit expression. So the right virus in the wrong cell leads to very poor transcription and sometimes no replication whatsoever. Regulatory proteins are divided into domains. They're composed of multiple subunits that contribute to virus regulation, and these involve things such as DNA binding domains, that is, sequences within these proteins such as zinc fingers or helix-turn helix motifs, or basic sequences that recognize DNA. Activator or repressor functions, which are involved in increasing initiation or tempering regulation. And these are frequently acidic in nature. They're glutamine-rich or proline-rich or isoleucine-rich. Interactors, things that bring the molecules together, that interact with other host proteins. And finally, multimerization domains, because many of these proteins don't act alone. They don't like to be lonely, they like to have partners. Sometimes they homodimerize, sometimes they heterodimerize, sometimes they multimerize, like SV40T antigen. So what are some of these virus transcriptional activators? Well, they can be autoregulatory molecules, like the SV40T antigen, or the herpes simplex virus ICP4 protein. Autoregulatory molecules are molecules that are synthesized, then go back and control the amount of expression from that gene. Some bind DNA. T antigen does it. The Epstein-Barr virus nuclear antigen does it. ICP4 from herpes virus does it. And E2 from the papillomavirus does it. Some bind host proteins. So when a herpes virus enters a cell, it has a protein that's contained within the tegument, the region between the core of the virus and the envelope, termed VP16, which is one of the most powerful transcriptional activators known in eukaryotic cells. And this protein interacts with host proteins, DNA, and we'll see this in a little more detail at the end of the lecture. And this facilitates entry and activation of genes. And some liberate host transcriptional activators. So T antigen binds DNA, it interacts with everything else, but in the course of interacting with the host, it also liberates a regulatory protein. Adenovirus has a protein that does a similar thing called E1A, and the papillomaviruses have a protein called E7, which is also active in that process. Now, proteins interact with polymerase II, and their purpose is to establish regulatory circuits, to define when various regions of a virus chromosome are transcribed. Again, there are positive autoregulatory loops. They alter the rate of transcription, and the virus proteins stimulate transcription. There are negative autoregulatory loops, and they repress gene expression. They suppress what the virus is doing. And sometimes that's important so that the polymerase can move to another promoter. And the most interesting feature of this whole thing is the generation of transcriptional cascades, where transcriptional units are activated in a fixed sequence. So sequence A, B, and C are turned on first. They may produce a protein that's involved in turning on sequence D, E, and F, etc. And we'll talk about that a bit. In the positive autoregulatory mode, a gene is transcribed, the viral messenger RNA gets translated, and protein X goes back and controls expression of gene X. In cascade regulation, gene X is transcribed, a protein is made, and it goes back to the viral genome, and it turns on another set of components that can be responsible for controlling transcription or for synthesizing virus DNA, remembering that virus DNA replication is an important control element in regulating the synthesis of late gene products. So, a transcriptional cascade allows for transcription of viral genes in a reproducible and a temporal sequence, again going back to the fact that some virus gene products, when elaborated at the wrong time in infection, will destroy the replication cycle. It allows for first transcription of immediate early genes and then later early proteins, and the later control of late genes. It ensures coordinated production of virus DNA templates and structural proteins, and it frees the templates from repressors. So early on, we believe that the late promoters are not seen because those particular sequences are suppressed. They're repressed by host they're repressed for transcription by host polymerase. But once you have a large number of templates, you now can saturate out the repressors, you free that DNA, and you allow those genes to be transcribed. So let's just look quickly at three different kinds of viruses. The polyomaviridae, SV40, where the viral DNA comes in. You'll remember that the origin is situated between the promoters for early and late transcription. And when T binds, not only the first transcripts that are made are the early transcripts, and they encode the T antigen. And when T binds to the viral DNA around the origin, what it does is allow for the initiation of DNA synthesis, and it relieves repression of late gene transcripts. Adeno enters the cell as a linear molecule, and the first gene to be transcribed is a gene that encodes products called E1A. And these are actually two different gene products that are derived by splicing, and I'll show you that in a bit. E1A is then involved in relieving repression by the host and allows the expression of early gene product E2. And E2 is the polymerase, the adenovirus DNA polymerase, and we've shown you that that's involved in DNA replication and a variety of other early proteins, and they lead to anti-repression and relief of promoter occlusion, and the synthesis of 4A. And 4A is a protein that accentuates utilization of the major late promoter and allows for the production of structural proteins. Herpes simplex virus does a little bit differently. Instead of being immediately transcribed, it introduces a virus protein, VP16, along with the herpes simplex virus genome. VP16 recognizes, in concert with host proteins, promoters for the immediate early gene family and allows for initiation of transcription from these gene promoters. They subsequently produce protein products such as ICP4, which go back and regulate immediate early gene transcription. They suppress it, but they also go on and activate early genes. The early genes, just like in adenovirus and just like in SV40, are replication proteins. They're responsible for replicating the virus genome, which results in large numbers of copies of DNA, relief of promoter inclusion, and allows for expression of late genes. Now, SV40, which is 5 kilobase pairs in size, has 2 units of transcription that result in 6 proteins. Again, please don't memorize numbers, just know the fact that there's an early and late sequence. There's a heavy reliance of SV40 on the host to get started because it doesn't bring anything in with it. It just brings in a chromatinized template. and it's splicing that generates multiple RNAs. Adenovirus is considerably larger. There are 8 transcription units. They encode 40 proteins, but there are a minimal number of transcriptional regulatory sequences because of splicing and the utilization of splicing to generate many of these proteins. The herpes viruses are about 150 kilobases in size, though they range from about 120 to 250. And they code anywhere from 80 to 120 different proteins. But it's the virus-specified transcriptional activators that are responsible for controlling the regulatory cascade. And remember that some auto-regulate, such as T antigen and ICP4, and others just activate, such as E1A. T and ICP4 are DNA binding proteins. E1A is not. Here's a picture of the SV40 chromosome as seen in the context of the transcription units. So the early transcription units encode a couple of proteins that are made from a single messenger RNA. And it's their differential splicing that leads to the formation of 1, 2, 3, 4 different RNA molecules. And you'll see that these RNA molecules overlap to some extent. They have a common 5' end, but they encode different proteins. Similarly, the late transcription units encoding structural proteins VP1, 2, and 3 are encoded by RNAs that also overlap, and they are derived by splicing. So it's splicing that allows for expression of various early and late SV40 transcription units. How does T work? Just the way it works for DNA replication. It binds to the origin as a hexamer, and it's that same binding that's used for transcriptional regulation. It results in dampening expression from the early promoter and activating expression from the late promoter. As a result, early transcripts are decreased relative to the late transcripts. And that's how you get more late messenger RNA and generate the large number of subunits that are required for putting together virions, structural components. Again, here's T. Early transcripts are made. They express T antigen, goes back to the same sequence, turns off or dampens early expression, and activates the late sequences. What about the adenovirus transcription regulation? That's governed by 3 virus proteins and DNA synthesis, and that's what's required to regulate adenovirus transcription. E1A is necessary for transcription of all early transcription units. E2, an early protein, is required for DNA synthesis and entry into the late transcriptional phase. And it results in increased initiation from the major late promoter, in part as a result of expression of the protein called 4A2, which enhances late gene transcription. Now, what is E1A? E1A is the first family of genes, family if you will, but it's the first gene that's transcribed during adeno-infection, and 2 proteins are derived from it, one of 289 amino acids, one of 243 amino acids. And they are identical except for the fact that the larger one contains a small region here called CR3, which results from differential splicing. So a splice occurs further down the template, but is juxtaposed to the same acceptor sequence so that the carboxy terminus of these 2 proteins are identical. The CR3 domain allows this protein to act with something called mediator 23. And mediator 23 is a protein that's part of the RNA polymerase holoenzyme. So you can immediately see how this may be responsible in part for controlling initiation of transcription from virus genomes. So how does this guy work? Well, the smaller guy interacts with a bunch of host proteins, the most interesting of which is RB, or the retinoblastoma protein. E1A does not bind DNA. It does bind these proteins that are involved in stimulating complex formation, and it interacts with RB and another protein called P300, which is a histone acetylase. But let's look at RB in more detail. In a normal cell, RB binds a family of proteins called E2F. And that's illustrated here as E2F plus DP1 because there are 6 or 7 different members of this family. But E2F is an important transcription protein. And when it is sequestered by RB, it itself can still bind to DNA, but it can't activate transcription. And it doesn't activate transcription because RB keeps it inactive. When E1A is expressed and accumulates in cells, what it does is binds in a site that's just adjacent to where E2F binds. And it liberates E2F as a result of that. So there's a conformational change in RB that allows for exchange of E2F for E1A. You now release E2F. E2F finds its way to the adenovirus chromosome and initiates transcription from the early gene family. So this is how E1A regulates expression of early genes from adenovirus. Now for something a little bit more complicated. And this is a schematic of the herpes virus replication cycle. Try to think of it as something not too simplex. So we have a virus that comes into a cell, and when the virus membranes fuse with the membranes of the cell, they release a protein called VP16, along with some other host proteins. VP16 binds to a protein called host cell factor, and it enters the nucleus. The capsid goes along a series of microtubules and pushed by dynein and enters the nucleus, where virus DNA is released. The linear form of the virion genome then gets circularized, and immediate early gene promoters are recognized by VP16 in the context of host cell factor and other host proteins. That allows for expression of immediate early proteins, and there are a number of these, and they get made in the cytoplasm and they get transported back into the nucleus. They in turn turn on the early genes. The early genes get expressed, get translated, come back, and they're responsible mostly for replicating virus DNA. Virus DNA replication allows for the expression of late gene products. Late gene products allow for production of capsid proteins, which come back into the nucleus and capsidate virus DNA, and then bud out from the nuclear envelope, acquire other virus and host proteins in a tegument, and in another process which involves re-envelopment, form a complete virus structure. Pretty complicated. So, the herpes virus regulatory cascade is initiated by VP16, a virion-associated protein. This protein, in collaboration with other host proteins, activates immediate early virus transcription. Immediate early virus proteins control transcription from all virus genes. So they are required throughout the infectious cycle. If you have a temperature-sensitive mutation in one of these immediate early virus genes, and you raise the temperature and inactivate that protein so that it can no longer do what it normally does, you stop transcription. So these are master control units. They're involved first in the expression of the early genes and subsequently DNA synthesis. Later, after DNA synthesis has occurred or is going on, you see expression of what are called delayed late and late genes, and these are DNA dependent. And finally, you bring the whole replication cycle back to the starting point because you package VP16 into the new virions. So it's an important component of the tegument, and it's an important structural element for making infectious virions. What is VP16? It's an acidic activator. So it's a protein that has a potent carboxy terminal domain that is acidic in nature. Like E1A, it does not bind DNA. And yet it requires a very specific DNA sequence called the TAT-GARAT motif in virus promoters. These TAT-GARAT motifs are found exclusively in immediate early genes. This virus protein associates with two host proteins, host cell factor and OCT1. OCT1 is a ubiquitous cell factor present in almost every cell type. And it's this quaternary structure that forms between VP16, the Tat-Garat motif, host cell factor, and OCT1 that provides promoter specificity. The interaction of these three proteins with DNA stimulates initiation and elongation of transcription. And again, there's specificity for immediate early promoters because of the presence of this DNA motif in those sequences. Now, how does it work? OCT1, a homeodomain protein, as I said to you before, is fairly ubiquitous in cells. The Tat-Garat motif contains within it a sequence called the octamer binding sequence that is recognized by this protein and the Garat moiety. So in a cell, when the virus chromosome enters, the immediate early genes are first recognized by OCT1. OCT1 binds and sits there stably, doing not much. OCT1 is a cell protein that was first identified as being important in transcription in lymphocytes, but it's been found to be fairly ubiquitous. When VP16 comes into the cell, in the cytoplasm, it acts with HCF. And that leads to a conformational change. And it allows recognition of the Garat motif in the context of OCT1. So if you deplete OCT1, you don't get the interaction of this binary complex with DNA. You need the sequences, you need both components of the sequence, you need OCT1, and you need VP16 to act with host cell factor to establish this quaternary complex. And it's this quaternary complex that contains this very powerful activation motif and results in generation of initiation complex. Now you can play games with this molecule, and you can substitute the amino terminus that doesn't have the activation domain with a DNA binding protein. So if you add a DNA binding protein and you add the activation domain to it, you can now place this molecule down onto any DNA that has that DNA binding sequence and activate transcription from it. And that's been done for a lot of genes in yeast, and it's one of the ways in which this was characterized. Now, one of the things that we have to talk about in a little bit of detail, but not too much, is export. And what happens and how do you get primary transcripts from the nucleus out into the cytoplasm? And remember that a primary transcript does not become a messenger RNA until it's exported. Things don't get translated in the nucleus. Export is usually accomplished by host proteins, and they use nuclear pores to exit. What we do know is that there's a protein complex that forms and marks mature RNAs for export, and that's assembled during splicing. And these protein complexes, which are represent a large number of different proteins, interact with things called exportins, whose role is to shuttle between the nucleus and the cytoplasm, carrying RNA as their cargo. So that's what the exportins do. How do you get an RNA out of the nucleus that doesn't have a splicing signal? Well, that's where viral proteins come in, and retroviruses, herpes viruses, and orthomyxoviruses produce unspliced RNAs that are exported for translation or virion assembly. So how do they do it? Well, let's talk about ICP27, which is a herpes simplex virus protein. And its role is to export single exon viral mRNAs. That is, RNA molecules that lack an intron. These RNAs contain signals that promote their export. And these RNA signals are recognized by motifs in ICP27 such that it now acts as a charon. That's the guy who crossed the river Styx, those of you who don't know, to ferry most HSV single exon mRNAs to the cytoplasm. So there's a very specific interaction of a virus protein with specific RNA sequences and the host export machinery to take these RNAs from the nucleus, bring them into the cytoplasm, where they subsequently get translated. ICP27 gets released from the complex and goes back into the nucleus. To do that, it has to have a bunch of motifs. Every protein has a motif. So what is ICP27? It's an interesting protein because it has a lot of activities that are very important to herpes virus replication. It's an immediate early gene product. The virus cannot replicate without it, so it's essential. It shuts off the host by interacting with splicing proteins. Why is that? Because herpes virus only has 2 proteins that are derived by splicing in the whole genome. It's a nuclear phosphoprotein. It also happens to be methylated in a bunch of arginine residues. It has a leucine-rich nuclear export sequence. And that's the sequence that's recognized by the host machinery to bring the molecule out of the nucleus. It turns out that it can actually interact with itself as a dimer or fold back on itself. It has an NLS, which is a nuclear localization signal. You've got to be made in the cytoplasm, and then you get brought into the nucleus. When you find your RNA cargo, you get shuttled out, and you use the NES. And that's why these are called shuttling proteins, because they move from the cytoplasm to the nucleus and back out to the cytoplasm. The protein is required for polyadenylation of these intronless RNAs. And as I said to you before, it shuttles. What is shuttling? In the course of virus replication, you can demonstrate that this protein, under certain conditions, and this was done using a temperature-sensitive mutation, will sit in the nucleus. Under this condition, the virus does not replicate. But if you shift the temperature down and allow the virus to replicate, then what you see is that the protein goes from the nucleus, and some of it accumulates in the cytoplasm. And we know that that's not de novo synthesis because we can inhibit de novo protein synthesis using protein synthesis inhibitors such as cyclohexamide and still demonstrate that protein moves from the nucleus out into the cytoplasm. And it's this movement that's essential for generation of translatable RNAs in herpes-infected cells. All right, so let's finish with something a little bit more simple, and that's the adenovirus late gene expression. And sometimes it's true that DNA replication is sufficient to generate late gene products. And that's probably a lie, okay? But that's a simple statement, and what it means is that we don't really understand for many viruses how late gene expression is controlled. But with adenovirus, we do know that when this gene product is transcribed, that what happens is you increase viral DNA synthesis, and this titrates out a cell repressor. 4A then cooperates with another viral protein to stimulate transcription from the major late promoter. And what you do is you get temporally regulated expression and termination of transcripts at different times. So early in the infectious cycle, you transcribe from the major late promoter, and it gets truncated. Things stop somewhere in here. And at late times, you get elongation and processive transcription that moves through the genome and allows the formation of that large single heterogeneous nuclear RNA, which subsequently gets spliced. So what did we learn about today? That transcription is complicated. It remains something to be worked on. Control is at many levels. There can be control from the host. It can be controlled by the virus. Things are temporarily regulated. They're coordinately regulated. But generally speaking, it's the interaction of host and viral gene products that regulate transcription. So there is a symbiotic relationship, of course, between the virus and the host. And there is, of course, the parasitic phase where the virus is taking what it can from the host to augment its own transcription. And finally, viral gene expression is coordinately regulated in a temporal manner. Thank you.